Welcome to an introduction to HPLC, overview and benefits. This training module is designed for practicing scientists in intermediate level, focusing on pharmaceutical analysis. I'm Michael Dawn. Today's agenda will cover: first, what is HPLC, including a brief history and its separation mechanism. Second, why is HPLC so useful? Explaining the benefits with a pharmaceutical analysis case study. Let's define some terms. Liquid chromatography, LC, is a separation technique where a sample's components are distributed between a mobile and a stationary phase. High-pressure liquid chromatography, HPLC, emerged in the 1960s by using high-pressure pumps to move the mobile phase through small particle columns. UHPLC, or ultra-high-pressure LC, is a modern platform with pressures over 15,000 psi and lower dispersion. Here's a brief history of HPLC. In 1906, Mikhail Tsvet coined chromatography. In 1941, A.J.P. Martin and R.L.M. Singh published on partition chromatography. In 1966, Csaba Horváth developed the first HPLC equipment. In 1969, Waters Associates introduced the first commercial HPLC. The 1970s and 80s saw advances in bonded phases in UV detectors, enabling routine HPLC use. Continuing the history, in the 1990s, James Jorgensen published research on UHPLC. In 2004, Waters Corporation introduced Equity UPLC, the first commercial UHPLC. Since 2010, LC/MS has emerged as a tool for scientific discovery. Traditional liquid chromatography is a common purification technique in organic chemistry labs. Solvents are gravity-fed through glass columns packed with silica. Fractions are then analyzed manually. This diagram shows a schematic of an HPLC system. It has four major modules: pump, injector, detector, and CDS. Also shown are the column oven, waste, degasser, and mobile phase reservoirs. Here are examples of modern HPLC equipment from major manufacturers: Agilent, Shimadzu, Thermo Scientific, and Waters. Automated LC with precise pumps, auto samplers, and online detectors, and CDS. The chromatography process separates the sample mixture into components by distribution between a mobile and stationary phase. Distribution occurs via partitioning. HPLC is useful because of its high separation power and sensitive detection, applicable to diverse analyte types, and precise quantitative analysis. HPLC/MS is a powerful tool. Perceived limitations include no universal detector, lower separation efficiency than capillary GC, and challenges for regulated testing. Here's a case study: a stability indicating UHPLC assay of a drug product. It shows separation of the API and degradation products using a specific column and gradient method. In summary, HPLC delivers precise analyses of diverse samples operating at high pressures. HPLC is a common analytical technique using a liquid mobile phase and a column with stationary phase. For more information, please visit http://www.chromeclass.com or https://www.mwd-consulting.com.